Olá, bem-vindos e bem-vindas, um bom dia a todos, eu sou Denise Barbosa e estamos aqui mais uma vez ao vivo do novo estúdio da McKinsey em São Paulo. Queria pedir desculpas pelo pequeno atraso, a gente teve um probleminha técnico que agora já foi sanado. O McKinsey Talks já se consolidou como um espaço para conversas dos principais experts do mundo sobre temas relevantes para a agenda de negócios e o tema de hoje é o combate à Covid-19. As vacinas realmente irão salvar o mundo? Today, we are hosting Michael Conway, Senior Partner at McKinsey in Philadelphia. Hi, good morning, Michael.
Good morning, Denise.
And also, we have Tracy Francis, also a Senior Partner at McKinsey based in São Paulo office and leader of B2C practice for Latin America. Morning, Tracy.
Bom dia. Tudo bem?
Tudo bem, tudo bem. Então, vale lembrar a vocês aqui também, em português, que vocês podem fazer perguntas a qualquer momento. É só mandar um e-mail para mckinseytalks@mckinsey.com. Por favor, contribuam, a participação de vocês é fundamental. Today's session will be in English. Tracy and Michael, let's get started.
Sure. Thanks, Denise. Michael, thanks for joining us today on this topic that's, very personal and affects all of us in so many different ways, be it social, be it health, our families, our economics. So we really appreciate you joining us today. This question of vaccines from early on has been something that's been widely commented in the media, amongst our families, also in the scientific press. And in your article, you in the title of the article, you use the phrase, "Will the vaccine save the world?" Can you tell us a little bit about, what motivated the research and how you actually went about the research?
Sure. I think just to introduce briefly, led McKinsey's global public health practice for 12 years. And as people probably know, McKinsey does work in this area across a range of different players, from, private sector manufacturers who are making the technologies that you're using in public health to, the international funders and groups like the WHO, and also a lot of work with donors. So, we've got a big base of knowledge that's accumulated, and particularly in the vaccine space. So what we did is we looked across the pipeline and understood all the products, vaccines that were in development. We did modeling, typical McKinsey, looking at things like, attrition rates for the pipeline, the impact of newer technologies versus older technologies, likely uptake, the interaction between, what a coverage rate will eventually be and the efficacy of a vaccine and how that will impact the spreading of COVID. And then we did interviews with experts. And, between all of that, we felt that we had a perspective that was worth sharing around the potential impact of COVID vaccines on the epidemic. And then in the article, as people may have seen, we talk about the likely timing and quantity of vaccines. We reflect on the drivers of the value for society of a vaccine, and eventually what it will take to bring them into the market. Right. I see that my picture, I think, has disappeared, but you guys can hear me. I'm trying to figure out what happened there.
Sure. Yeah. Today appears to be the day of technical hitches.
Yes.
So talk us through a little bit the broader context here, right? The history of vaccine development and timing, to give us a bit of a context as to what the world scientific community is trying to get done.
Yeah. So, I thought about that question, and, if you think about a normal vaccine development program, you spend probably 3 to 5 years just understanding the pathogen that you're dealing with. And, in the case of COVID, some of you guys have probably noticed that the spike protein is the one that everyone talks about. It's the protein that you see in pictures sticking out from the virus. And that's an obvious thing to go after because it's part of what allows the virus to attach to a cell. But there are other proteins. And sometimes it turns out that some proteins in a virus mutate more than others. So in normal viral development, you would spend years thinking about this question of which protein, or which, it may be a carbohydrate too, but typically it's a protein, is the one that we want to go after. And we didn't do that here. We've gone pretty quickly to the spike protein. It's one of the things we reflect in the article. If that did turn out to be a bad target, literally 70% of the vaccines are using that. So that will be a systemic failure. But it doesn't look like that. At this point, we have enough evidence that it seems like the spike protein was a good target. You normally then do phase 1. You spend time, looking at all the data from the 10 volunteers who took your vaccine. You follow them for maybe a year, so you really have pretty good data from phase 1. We're not doing that here. We've jetted right through phase 1 into phase 2 in many situations. Very quick phase 1. We know that you don't fall over when we give you the vaccine. Then we're doing phase 2. The phase 2s are very large. It's on the order of the size of any phase 3, and that's the standard that the FDA put out. But normally, you would follow those patients again for a couple of years. And, one of the reasons you do that is you want to know, does the protection last over time? Because with a new vaccine, you don't typically know. And you guys have probably seen in the media that, we've got some reinfection cases for COVID, and there's, just debate there going on about how protective is even getting the disease. And obviously vaccines are usually a little less good, and in a few cases they've been better, but often a little less good than natural disease. The early data again shows that the vaccines are causing antibody titers that are higher than natural disease. So that's one of the reasons people are feeling, again, more confident. And while there are some rare cases of reinfection, the belief is that it's probably not common. There's just some weird cases that are happening that we're picking up. And then you probably spend a year or two, Tracy, dealing with the regulators. So all of that could be, 7 to 10 years. And here we're going to do that. It looks like in October, November is when the early guys, the Pfizers, Modernas of the world, are saying that they will have a data file that they can start to share with the regulator. The big thing, just the last point, when people talk about emergency authorization, the thing that we're going to do is we are not going to have followed all the patients for 3 years from the phase 3, but we will have 20,000 patients data. So when people talk about we don't have enough data, I think you have to look at that in context. We will be pretty sure that there's not a safety signal, because normally that comes early. So you're now into the very small set of weird safety signals that might show up, with very small groups of people over time, but that's not the normal thing. And then, we will have pretty good data on efficacy. You won't know the 3-year data, of course, but, we will know that, for most of those 20,000 people, 1 or 2 months after you got the shot, you still have pretty good antibody titers. And again, in the usual world, we would have waited for 2 years to know what you have. But on some level, I don't think that's the thing that everyone should be worried about in terms of the population, if we use an emergency use authorization.
Got it.
Sorry.
No, not at all. So in your article, you talked about how the fastest vaccine to date, I think, was mumps, right? Which took 4 years to develop, right? So here we're going for like a year, I guess a little bit less than a year. So talk to us about, what are the scale of resources that are being devoted to this process, and, reasons to hope for success here?
Yeah. It's really just completely unprecedented. I think the biggest thing that you immediately see is versus recent history, you have all the major vaccine manufacturers and many biotechs and small developing country manufacturers, smaller developing country manufacturers who are pursuing vaccines. You have collaboration on a scale that we've never seen before. So the vaccine manufacturers are sharing information across the industry. The spike protein that I talked about, people have been very forthcoming about what they're doing and what data they have. And that's just something we haven't seen before. The money is probably, I don't think anybody really totally knows the number, but let's say it's on the order of 8 to 10 billion. which, is an unprecedented, quick investment. So that's really probably the biggest thing. And then, of course, if you look at any clinical trial center, which is just interesting, there are just so many trials running, and most of the centers are doing multiple trials, the really big clinical trial centers, which is, again, that wouldn't have happened historically. They usually have to pick something.
Yeah, I think we have a slide here that looks at the scale of the number of candidates here. Let's see. You're back. You've got your photo back. Got your image back. So we can see here there's 250 candidates in development. So just give us a little bit of color on what's where. And also, you've got these technology platforms, right? Perhaps you can talk us through a little bit. What are the different technologies that are in play for lay people?
Yeah. So 250, I mean, normally, again, for like pneumococcal vaccine, which many people know about, that's Prevnar. That vaccine, 2 companies eventually brought the vaccine to market. If you count the polysaccharide version, maybe there's 5 in the world. So 200 is a big number. And when you look at this chart, just starting from the bottom, so the protein subunit, that's the normal approach for a vaccine, right? You've taken the spike protein, you take part of the spike protein that you think the immune system can recognize, and you then produce that protein using some biologic production method. So that's the way we've all been historically making vaccines. The DNA, RNA, and viral vector, those are all new approaches. And everybody was always excited about these approaches because they're easier to manufacture. And in theory, we'll be able to scale very quickly using these approaches. The way they work is actually interesting too, because in the other versions, you're using some kind of cell to manufacture the protein that you want the body to recognize. Occasionally, the protein's not exactly the same. Things like sugars that get attached to the protein is the kind of thing that is sometimes different in a different organism than a person. When you use the viral vector, you're actually taking a virus, and AstraZeneca, the Oxford one, is a chimp adenovirus. So that's effectively a cold virus that impacts monkeys. And you've made it so it can infect a person. So nobody should have antibodies because you shouldn't have ever gotten it before. And that is basically going to insert the protein just like a virus would to then make the protein in your cells, which is the antigen, the spike protein. So, it's really interesting. It will be exactly the same, presumably, as the spike protein that's made by COVID. The RNA and DNA are doing almost the same thing, with the difference that they're delivering the DNA or the RNA through some other vector. It could be a small encapsulated bit of RNA, but effectively you're going to end up with the same thing happening. Once it's in the cell, it's going to get transcribed into the protein. You're going to make the spike protein in human cells. So in fact, all of the leading products, except for Novavax, are using a new technology. And it's one of the things that people obsess about a little bit. On the other side, viral vectors is all the gene therapies that you guys hear about are viral vectors. There are lots of viral vectors that are, lots of clinical trials have been done. They're on the market. There are even more RNA and DNA therapeutics. So while it's new for vaccines, it's not new for pharmaceuticals. And so I don't probably feel as concerned. I think it will fail not so much because the technology fails, it will fail because the spike protein, turns out to be wrong. The last piece, the virus-like protein, is just you take the antigen and you stick it onto something that looks like the virus, which just helps the immune system to see it. Live attenuated is actually a virus, right? That's giving someone a COVID virus that has been adapted in the lab to be not disease causing. And the fear always with live attenuated is what if it would become unattenuated again. But again, long history with live attenuated viruses, or you get into an immunocompromised population and somehow it just doesn't get wiped out. But that's what polio vaccine is. Again, it's another area. There are vaccines, and inactivated is basically where you take the virus and you kill it. The advantage of inactivated is that you have everything. Whatever was in the virus is in the inactivated slurry. The disadvantage is that at least sometimes inactivated, because there's so much stuff in there, just causes your immune system to have an extreme reaction, which is usually not a problem other than the fact that it's uncomfortable for the person who got the vaccine. So that's kind of the picture.
Okay, so I think I sort of understand. Lots of information here, but one of the things I very much liked in the article is you guys made a matrix that sort of says, okay, of these 250 vaccines that are in development, what are the sort of chances of success? And so perhaps we can put that chart up, and you can talk us through how we should think about this.
Yeah. So what we did here is, look at two dimensions that are going to make a difference on the probability of success. So one is, at the end of the day, if you were to look, you can find, roughly 260 candidates that people say they're working on. And, clearly some of those are already things that people probably have decided don't have distinctive advantages. They're not early enough. They won't ultimately develop them. It still is pretty shocking that there are 56 candidates that are going into clinical trials. I talked to you guys, normally you might have 3 or 4. So we're big. So the first thing we did is we said, well, will all those, candidates, even if someone says they're entering clinical trials, they don't necessarily start the trial. They can work their way up to a point and stop. So let's imagine that either just the guys who are in the lead go forward, or you actually see this big pool coming forward. And then the question is, the new platforms I talked about, they're platforms that have been used in healthcare, like RNA, DNA. They haven't been used in vaccines. And so some people would argue, you should handicap them because maybe they won't turn out to be great for vaccines. And so we basically took a small, change there in our thinking. But the bottom line is, it is very likely that we will get out of the current efforts, at least 7 vaccines, and it could be more. And it's also, if you look at the timing, the fourth quarter, first quarter, looks very positive. And even this morning, Pfizer came out and said, we're on track for end October, having our data file. So, there's nothing that's happened that throws you off from believing that we will have vaccines. We'll have them, with emergency use approval. The trials won't be done, but there'll be 20,000 people who've gotten the vaccine at the end of this year for multiple products.
Excellent. So we're talking about, potentially having a vaccine end of year. Of course, no one has a crystal ball, but based on the analysis, I understand that that's a likely outcome. What are the hurdles, the sort of non-science hurdles between having the data file and getting the vaccine to the population or some part of the population?
Yeah. Well, so this is something we've started to work on, and there's some important hurdles here. You guys are all business people. The supply chain is challenging, and it's challenging because one thing I didn't mention, the RNA vaccines, at least today, are normally stored at -80, which is basically, I guess, dry ice, liquid nitrogen, very cold. So there's a lot of work going on trying to figure out, well, could we have them at -20 for a while, or even, 4 to 6 Celsius is a normal vaccine, because that's a normal freezer. So, I think we'll see what we're going to find. Our normal refrigerator. We'll see what we're going to find, but that's going to be quite challenging. And in the U.S., a lot of investment has already gone in, because these first generation vaccines will require that kind of supply chain to trying to figure that out. And then there's the question of, how is your program actually going to be designed, and who are you going to be targeting? So, a lot of people say healthcare workers, as a first target group. That one's a little bit easier, because that's like an employment situation. You know where they are, they're all in one place. But then when you get to, at-risk populations, the history of at-risk populations is very bad in vaccinations, because you're going to tell me that I should do everyone above age 30 who's got diabetes or hypertension or, blah, blah, blah. And then, how do I find them? how do I get them to come at a specific time to a specific place, or am I going to try to go to their place and vaccinate them? So I try to get them to come into a medical practice, but then I've got to split these doses that I'm storing at -80. So that part of it is just very challenging to think about. And then there's the public relations part. And you guys have seen in the US, all around the world, this has already created this, concerns. Some of the concerns are legitimate about safety. Do we have enough data? And I think that, I tried to explain how I view it, because I'm trying to be, not a zealot, but a sort of clear view of this. I think we'll have 20,000 people who've had, 2 months of experience, and, that leaves a little more residual risk than normal, but not crazy, versus the risk that a lot of these populations face in COVID. And the other thing, as I've talked to my friends, is, okay, and then, by the first quarter, the second quarter next year, we're likely to have 1 million people, and that's unprecedented. And the point at which, you're deciding whether or not to vaccinate your child, at least in the US, ranking of how we're going to do this, is like third quarter next year. By then, you've got tons of data on safety. So, trying to give people a little bit of perspective here, so they don't focus on the decision they're not actually trying to make, and then get very anxious about it, is just a delicate communication challenge that we've never faced before, and with probably more concerted counter messaging than you faced before. And then the program management is just challenging, Tracy. You're trying to move very quickly, and public officials historically take long periods of time to think about how to set up a program like this. And they run pilots and they think about it, and we don't have time to do any of those things.
Yeah, well, thank you for your candor. I wanted to address two other things. We started a bit late, so we don't have a huge amount of time, and I do want to leave a bit of time for questions. But I think there's two other topics that are really interesting, right? One is, what is the overall value of this vaccine to society? And of course, it depends on how the vaccine actually comes to market. So that's the first thing. And then the second thing is, I wanted you to talk about how the vaccine fits into the broader context of other measures that that we can be taking. So talk us through those things, and then we'll take some questions from the audience.
And I can do those too, because to me they're connected. So if you think about where we are in our response to COVID, there's 3 big priorities that we think about from my side. So one is the preventing transmission. And, it's an area where we've got lots of data, and we need to start to look at it and think about it and refine our approach. And some early work that's just coming out from McKinsey, but other people, and it's not public, but just being done, some of it's public. People are trying to look at things like mask requirements, and, how much does a mask requirement make a difference? The answer is a lot. And then they look at other things like travel restrictions, or can you go into a restaurant? And, this is, of course, as you guys know, data is messy. These are small decisions in a big context. So, take it with a grain of salt. But it looks like, having restaurants that are, widely spread with controls to ensure that employees aren't sick is a big step. Taking the step of closing the restaurant doesn't get you a lot more juice out of the lemon. So I think the world will just keep working this and getting smarter, and we are getting smarter, and making lockdowns and restrictions less onerous. The second point is treatment. You guys have seen remdesivir, everyone talks about it, but there's also steroids. There's lots of approaches to treatment. The case fatality rate in the US has dropped dramatically, and it's very difficult, and I keep pushing my colleagues to get the actual numbers. But, some people would say it's 98% down. I think that's not true. That's crazy. But it's down a lot from where we started. And that will continue to happen, right? And so those 2 things are really important. We can reduce transmission through non-medical means, and we can, do a better job when people do get sick of not having them have bad outcomes. Then, vaccines, and frankly, monoclonal antibodies, which I also believe in, prophylactic monoclonal antibodies, that's what Synagis is very important. And that's where the value to society comes from, because in a world where we do a great job on 1 and 2 that I just talked about, 3 is less interesting. In a world where the vaccine has less efficacy, like a 50% efficacious vaccine, is much less impactful on the spread of a disease than a 97% efficacious vaccine. And most people, tend to talk in 50 to 80 on coronavirus. That's just consensus. I don't think anybody really knows what the answer will be in the end. At 50, the value is less. And so, if the disease, and the last point is how quickly is the disease really spreading, which is amazing to you guys. But, all we know in the US is that the number of cases that have been confirmed with a lab diagnosis, the actual number of cases is still debated. We're still doing studies and getting different numbers on, is it 10% of the people in the country that have had this now, which is about the kind of number that a lot of people talk about, 6 to 10, somewhere in there. But, in some places like New York City, some people think that, in parts of New York City, we're already at 50%. And that, of course, makes a big difference. If 50% of the people in Brooklyn have had COVID, then clearly introducing the vaccine, assuming that there's durable, and that's the question, but assuming that you have durable immunity is not going to have the same impact. So what we try to do is lay out the factors and sort of say, look, if you believe this, then the value of the vaccine is higher. If you believe that, it's not. In any world, the vaccine is very valuable. And, so, any world that seems probable today, if it turned out that there are 50 COVID cases for every measured COVID case, then we might not need a vaccine because the world will all have had it. But I don't think the data that we do have suggests that's not true. Yeah.
Super. Michael, as I said, I want to make sure we have time for some questions from the audience, but I think that was incredible, right? A tour of, why we should hope for success, a tour of what else we can be doing, and a bit of an understanding of what the value of the vaccine to society is. So Denise, back to you for some questions.
Okay, thank you, Tracy and Michael. So now I'm going to address some of questions sent by our audience. So Michael, the first question is, will the novel vaccine platforms and technologies being tested have an impact for other research, even if they fail for COVID vaccine?
Yes, that's a really exciting part for those of us who work in vaccines. The reason that people have always been excited about RNA or DNA is what I said to you guys. It's that you're producing the protein or the antigen in a human cell, and there's a good chance that you actually will end up getting a better, more natural immune response. So people have always been excited for that reason. The viral vectors is probably less of a technology for vaccines, because the problem is, even with the Oxford, we don't know yet, but it is highly likely that your immune system picks up that chimp adenovirus, and therefore the second time you give someone the chimp adenovirus, which is the problem that we have with gene therapies as well, the body wipes out the vector before you get the impact. So for vaccines, I don't think most people think viral vectors are going to be a big solution, but the DNA and RNA has been for years talked about as an exciting approach.
Other question: when does the window of innovation for COVID vaccine close? After 7 to 9 vaccines are available, will others continue to pursue it to make more options available?
Yeah. So, here I look at flu, and, probably 10 years ago, when I would have worked in flu, everyone would have said, it's done, this is a dead market, there's nothing that can be done in flu. And, that was a world where, for older patients, and it's likely to be a challenge here too, they might only get 50% efficacy. And the thing that no one ever talks about in flu is, it wanes pretty quickly. So by the end of the season, if you get your flu shot in September, by March, you're pretty low, efficacy versus where you started. So there were unmet needs, right? People could see that there are ways to make this better, but still somehow everyone said, yeah, but it can't, nothing's going to get better. But then now we have HD flu, which is high dose, right? You give someone 3 times as much antigen, and shockingly, it turns out that they get a better immune response. And, in March, they're much better than they would be if you gave them the traditional. And then, you gave them adjuvants, which is just something that makes the immune system recognize and go harder. And that turned out to be better too. And now it also turned out, because we were making flu vaccines in eggs, that when you make flu vaccines in cells, it is a better protein that's closer to the natural protein. This is the whole point that we all know, right? And so the immune system's better, and the cell culture vaccines in the last season had better efficacy. So certainly within COVID, there is a belief that this first generation of vaccines are likely to be not the best that we can do, and therefore some of the ones in the pipeline will still be successful. And the other question is cost. And you guys saw that the Gates Foundation just agreed to work with Serum Institute of India. And the truth is that making large quantities of doses cheaply, it looks like AstraZeneca is making big commitments, but that has not historically been the forte of the Western pharmaceutical industry. And, I use the example of Menafravac, which was a MenA vaccine that Serum made. And I was around when that was brought up and it was going to be less than $1 a dose. And the entire industry was like, that's impossible. Nobody can make a meningitis vaccine, which is a conjugated vaccine, for less than $1. And, Serum has been making it for less than $1 for probably 10 years. So, that is another form of innovation that will absolutely happen here, particularly if it turns out that we need to immunize every 5 years or every 2 years or some frequency. I'm hoping, by the way, that won't happen, because normally the second and third infection, you guys know dengue is the rare thing that's worse, but most things it's better. And therefore, we may not have to vaccinate the whole world multiple times here. But that, of course, would make a sustainable industry. There will be innovation for a long time.
Sure.
But if that happens, yeah.
And Michael, another question. What other work is being done on COVID prevention and treatment besides vaccines? Are there equally promising advances in treatment or other forms of prophylaxis? What would that impact the vaccine development?
Yeah, well, that's our value to society point, but but it will also impact the demand for vaccines. On the prophylaxis side, the most obvious other thing is monoclonal antibodies. And so Synagis is an RSV monoclonal antibody that AstraZeneca has been making for like 20 years. And we know that it has very good efficacy. It's used with premature infants. It's expensive, right? Even, when you say that this will be done at cost, it will still, that's an expensive way to do it. And there are monoclonal antibodies now that have 6 months duration. So they're modified proteins that the body doesn't quite recognize and break down. So that's good. 6 months is really good. So I feel like there is a chance that for part of the population, that might be the answer now. Like for a very old person, we often get very bad results with a vaccine, right? Their immune system is at this point degrading. So that's a solution that could definitely have a long-term part of the market there. It's also a pretty good solution, if you're really going to be hitting someone with a lot of pathogen, potentially a monoclonal antibody is actually better. So that's the big one. There could be things that are like immunomodulators that somehow amp up your immune system. I don't know. I'm not totally believing in that. In HIV, obviously, we're using antivirals prophylactically. So, of course, in theory, you could use antivirals prophylactically here. For a long time, people would have said that's not a great idea because there are side effects. But now, of course, with Trememba and a bunch of things in HIV, you guys know there are lots of people taking antivirals prophylactically. So I feel like that's possible. People haven't been going down that road. So far, more focused on monoclonal antibodies. There's a lot of monoclonal antibodies in development. And then also for treatment, convalescent plasma, which you guys saw, didn't have good results. I was just in a meeting where at least one senior, very savvy scientist said, I think what that really proves is that our data systems aren't very good, because we know from other viruses that convalescent plasma normally works. So it's a little bit surprising that it didn't, maybe it really will turn out that it doesn't. Lung diseases can be a little different. The immune system doesn't get into the lung that well. So, but we'll see what we see, but convalescent plasma would have been on everyone's list. And I still think it may turn out that it actually does work. And then there are antibodies that are being used also as a therapeutic, but that's similar to convalescent plasma. So I think it'll be interesting. If convalescent plasma really doesn't work, then antibodies. But then there's all the things you guys have seen. There are things that are tamping down the immune response, so you get less damage from your immune system, which are repurposed cancer drugs and other immunodiseased drugs. There's a lot of stuff in the pipeline. There's, a bigger pipeline, Tracy, than even in vaccines. Everyone who has anything that they think might work is testing it.
I think we have time for one last question, Michael. The analysis points to a potential capacity to produce up to 9 billion doses in 2021. What else is needed to make those doses reach populations on what will be a historical vaccination campaign?
Yeah, I mean, I think, the challenges that I mentioned are the same around the world, but bigger. And, in having worked in Africa in public health, you've got places where you don't actually know who your healthcare workers are. And I make the point frequently, we did work at one point on children, and like half the people are taking their children to private providers, but many of those private providers are informal. So are we going to reach the informal people? Because today, many countries ignore them, right? So they don't know who they are. So you'd have to actually go out and figure that out. And that actually creates challenges. One of the reasons you ignore them is that they're providing a service that your government wants to be provided, but you don't want to endorse them, so you don't track them. So it'll be interesting to see how people do that. And then there's just the nightmare, with polio, we ran into the problem. I was working in Nigeria, there's just constant swirls of information that it sterilizes you and things that aren't even, credibly within the potential. And then, the minus 80, like that feels pretty hard to imagine that we could do anything at scale at minus 80. Hopefully it turns out that you don't need minus 80. So for some of the vaccines, I don't think it's realistic. And even on the ones that aren't that, are you trying to do campaigns? Are you trying to do targeted? Again, if we were going to try to target high risk populations, it's hard enough in the US or in Brazil, to try to do that in Nigeria feels very, very difficult to imagine. So then are you going to try to do, age cuts, but these are the things, and then getting people moving on this. So you guys have seen that Gavi has this global purchasing facility. I think that has gone very quickly and is moving nicely, getting all the countries in the world to start thinking through. And part of that is that many countries, really don't believe they're going to have a vaccine even for another year. I think that's what I hear people saying is, look, Michael, it's a year, we've got time, we'll see what happens. And, that sort of is true, but historically you took more than a year, for example, to introduce pneumococcal. So, you probably should get going. That's kind of anyway.
Okay, yeah. Michael, thank you very much. Muito obrigada pela sua presença, Tracy. Obrigada também pela conversa aqui com a gente. E obrigada a vocês que mandaram perguntas, que nos acompanharam aqui nesses últimos minutos. Super obrigada pela companhia. Para conhecer a agenda completa do McKinsey Talks, acesse mckinseytalks.com. Lá vocês podem assistir aos episódios anteriores e na segunda-feira esse episódio vai estar lá. Tracy, você quer dar uma palavrinha final rapidinho?
Não, só queria agradecer. Many thanks, Michael. It was absolutely fascinating. We appreciate your time.
Thank you.
Thanks, Denise.
Obrigada. Então, voltando ao nosso assunto. Na segunda-feira esse episódio vai estar lá. E para quem gosta de podcast, esses áudios também estão disponíveis no Spotify. Muito obrigada. Tenha um ótimo fim de semana. Tchau, tchau.